Right. Hello, everyone, and welcome to today's SEDS Online webinar. As always, we would like to thank our sponsors at the IAS, which allows us to offer all of these resources free of charge. This includes recorded lectures, learning tools, and even virtual field trips. So take a look at the website to see all the new info. Today's lecture is by Dr. Anna Prohenka, who is a paleoecologist, paleogeneticist, and assistant professor at the University of Copenhagen. Anna received her master's and PhD degrees at the University of Oxford, and her research interests include understanding some of the key challenges in biodiversity conservation, including climate change, land use change, and other environmental stressors. So thank you so much for being here with us today, and I will hand it over to you.
Thank you, Simone, for the kind invitation. And also thank you. Yeah, it's a great to have the opportunity to contribute to this seminar series. So as the title suggests, my focus today will be on microfossil DNA. So I will basically introduce it as an heavily underused proxy, but it holds a great potential, not only for the field of ancient DNA, but also for the related fields such as evolutionary biology, paleoecology, and similar. So I will start with covering some of the key advantages of microfossil DNA, but also some of the main challenges of working with it. And then I will finish with discussing emerging opportunities for overcoming these challenges. To understand the unique value of microfossil DNA, we actually need to first take a step back and look at another proxy that has been in much wider use over the last 10 years. And that is ancient environmental DNA. So probably many of you have already heard about this proxy. In its simplest definition, ancient environmental DNA is DNA that we retrieve from ancient environmental deposits. And we can retrieve from many different deposits, many different environments. Some of the ones shown here, such as permafrost, terrestrial deposits, lake sediments, and marine sediments as well. And another important source is also archaeological deposits, archaeological sites. So irrelevant of the type of environment that we're getting DNA from, the basic principle is that organisms over their lifetime are basically shedding their DNA into the environment in the form of skin cells, hair, urine, feces, and other ways. And so this DNA then gets deposited in the environment. And if the conditions are favorable, such as dry conditions or cold ones, or ideally both, and also recent evidence suggests that also if the DNA gets attached to mineral particles or some sort of complex organic structures, for example, humic acids, that protects DNA from nucleases, which are enzymes that are basically degrading DNA. And so under these conditions, DNA can survive in the environment for a really long time. As our group shown here, even more than 2 million years. So the field of ancient environmental DNA has exploded over the last decade. It was named the runner-up of the Science's Breakthrough of the Year award for 2022. Also, in terms of the publications focusing on ancient environmental DNA, we can see a major uptick over the last 30 years, especially the last 10 years. And also we have Ancient Sedimentary DNA Society that is organizing meetings, biannual meetings, I believe, with the first one that was in Potsdam in 2023. So it is a fully fledged field. And it's also growing exponentially. And so what is the appeal of the ancient environmental DNA as a method? Well, arguably the biggest advantage is that it actually allows us to reconstruct ecosystems from a single sample using a single method. And that is very powerful. So if we have organisms on a landscape and they are depositing their DNA in some kind of archive, we can actually retrieve their DNA and identify what those organisms were. And that can be anything from viruses, bacteria, all the way to fungi and higher plants and animals. And so a good example of what ancient environmental DNA can provide is this study from 2021 by Wang and co-authors. So we basically extracted DNA from lake and permafrost sediments, more than 500 samples across the Arctic Circle. So you can see these locations here on the map. And they span the last 50,000 years. And this allowed us to actually reconstruct biotic responses to the past climatic changes at an unprecedented spatial resolution. And now another advantage of ancient environmental DNA is that actually it's quite efficient in detecting small populations. If we have a large population with a lot of individuals on landscape, we are likely to find both eDNA in the sediments that are still deposited somewhere nearby, but also to find skeletal remains. But if we consider a smaller population where maybe we have a few individuals on the landscape, such as in the case of declining and maybe close to extinction populations, then it is unlikely we're going to find skeletal remains of those last few individuals. But as we've shown, that actually we can potentially identify those last individuals through eDNA analysis. And this was shown again as part of the same study from 2021. So we took mammoth as an example. And you can see here the polar, Arctic polar maps. And you can see all the sites where mammoth presence has been confirmed, either through fossilized skeletal remains that are depicted with a circle, or through eDNA evidence, which is shown as triangles. So if we take the last glacial period, we can see there are many sites with the confirmed presence of mammoth, both through fossilized remains and through eDNA. But as we move to the Holocene, we notice two things. First, that there is a significant decrease in the number of sites where we have confirmed mammoth presence. But also in terms of skeletal remains, we are only seeing that evidence on the Wrangel Island, which is well known, one of the last holds of mammoth. But with eDNA, we were actually able to find additional sites elsewhere in Arctic, in Siberia, with supposed presence of mammoth all the way to the mid-Holocene. So actually with this method, with eDNA, we can start revisiting our understanding of the geographic ranges of some of these extinct megafauna and have a better understanding of the spatial trajectory of their extinction. And so in addition to also expanding the spatial scope of paleo studies, ancient environmental DNA is also pushing the boundaries of how far in the back we can actually construct whole ecosystems. So the oldest DNA that we managed to retrieve to date is about 2 million years old. And it was extracted from a site in North Greenland called Cap Copenhagen. That has been dated to 2.2, 2.3 million years ago. And based on the eDNA data from this site, we were able to show that this community, we're focusing on higher plants and animals, that these assemblages were actually non-analog. So it means that nowhere today on Earth we can find a similar ecosystem. And so these are some of the plant taxa that we have identified. So there were many of them, and many also of them have been identified through fossil pollen records and through plant macrofossils. But the ones that are in bold, and I apologize if it's not very obvious, those were only identified through eDNA evidence. And a nice example is populus or poplar, which is an important Arctic and boreal tree. And its presence in northern Greenland was not confirmed until this study that used eDNA. But the major finding really was with respect to animal assemblages. So first of all, you probably noticed that the list of animal taxa that we identify with eDNA is considerably shorter than plant taxa. And that's because eDNA is heavily influenced or shaped by biomass of the taxa they're contributing. And of course, plants have considerably bigger biomass than animals do. But nevertheless, we managed to identify several animal taxa, such as reindeer, geese, a type of rodent, a hare. But the biggest surprise was the apparent presence of mastodon, which is a member of the elephant family. And Cap Copenhagen is now the most northern confirmed site of its presence known to date. So this is, again, another example of where eDNA has revisited the geographic distribution of extinct animals. So I think that you would probably agree by now that ancient environmental DNA is quite a powerful proxy. It can do many interesting things. So it would be fair to ask why this talk, my talk today, focuses primarily on microfossil DNA. So what is the added value or the unique value that microfossil DNA brings to the table? Well, despite all its clear advantages, like any method, ancient environmental DNA also has some limitations. And to illustrate what is arguably its most significant limitation, I will compare here the kind of DNA data that we're getting from ancient environmental DNA versus macrofossil DNA. So ancient environmental DNA is basically a metagenomic sample. And this means that the DNA in it comes from many different species and often from multiple individuals of those said species. And in contrast, with the DNA that we get from macrofossils, such as bones and teeth, these are genomic samples because overwhelming the DNA that we are getting from it, apart from microbial contamination, belongs to not only a single species, but a single individual of that species. Now, thanks to some fantastic work in bioinformatics area over the last 10 years, we have become quite good in identifying what taxa contributed the DNA in a metagenomic sample that we get from the environment. And we do that by basically identifying which reads assign to different taxa using reference genomes of those taxa. So in the end, we end up with knowing that these reads or these DNA fragments belong to species 1, and these to species 2, species 3, etc. But there's one key information that we're actually not getting, and that is that we don't know how many individuals have contributed to the pool of DNA fragments belonging to species 1. Was it one individual or several or 100? And we are not able to say which of these DNA fragments that we retrieve belong to which individuals within that species. And so as a result, we are basically not able to build individual or single genomes. Why is there a problem? Well, because individual genomes, they allow us to study evolutionary and demographic processes at much higher precision. These are complex processes which include gene flow between populations, natural and artificial selection or domestication, to be able to track migrations of populations and their mixing. This is much better, much more precisely done with single genomes than with metagenomes. But of course, traditionally now so far, we have been getting metagenomic data from environmental samples. And the question is, can we get single genomes from environmental samples? And actually there is a way, and you can probably guess where I'm going. So if we look at a typical environmental sample, such as sediment sample here under light microscope, we can see that it consists of different components. So we have different mineral particles, we have amorphous organic matter, and we have these tiny fossils that we refer to as microfossils that are generally less than 1 millimeter in size. And in example here, these are pollen grains. Now, all of these different components have some DNA associated with it. So mineral particles, amorphous organic matter, and microfossils, they all yield some DNA. But the DNA that we are getting from minerals and amorphous organic matter, it's not the original DNA. So it's DNA that got released by an organism and it then got attached to these components. So it's a kind of a secondary DNA. In contrast, the DNA that we actually find inside of microfossils most likely belong to the organism that produced those microfossils. So in a way, like with the macrofossil remains, bones or teeth, this is primary DNA. At the moment, the way we are processing environmental samples standardly is that we take a piece of sediment of certain weight, then we introduce lysine matrix and we shake it vigorously. We completely destroy these different structures inside and release DNA, and the DNA from different components is all mixed up. But alternative way, and one that is actually used very rarely, is to actually isolate the targeted parts of the sediment, such as microfossils, and then do the extraction of DNA specifically from them. And why microfossils are a good source of single genomes? Well, the first thing is that a large number of different species produce microfossils. There's a huge diversity of them. So they range from single organism fossils, such as diatoms and coccolithoforms and foraminifera. They also can be reproductive units, such as pollen and fungal spores. And they can also be dormant units, such as bacterial spores in sediments. And on the other end of the spectrum, we are also having pieces of much larger organisms, such as pieces of leaves, of wood, roots. In terms of animals, we get shards of bones inside of sediments and similar. And a big advantage of microfossils over macrofossils is that they are several orders of magnitude more abundant. So in a single cubic centimeter of sediment, as many of you will know, we can get hundreds to potentially millions of pollen grains. And then comparably also to eDNA, microfossils can be found in a majority of depositional environments around the globe, from lake sediments, marine sediments, caves, ice cores, speleothems, etc. So they are ubiquitous. But a distinct advantage that microfossils have over eDNA is that in addition to genetic information, they also provide information about organismal traits. And many of you who work with fossils and with morphological analysis would be aware of that, right? So we can actually, and this work has been done for decades, where we are looking at changes in the size and the shape of different microfossils, such as diatoms, pollen grains, understanding how organisms responded to climatic changes. We also are looking at the structure of specific microfossils inside, for example, in pieces of leaves. We are looking at the density of stomata to understand the plant responses to changing CO2 levels. We also count pollen, and pollen counts on a more broader temporal scale can tell us something about the abundance of certain plant taxa. On a more finer scale, it can actually tell us something about flowering rates of certain plant taxa. So that's on the morphology side. But then on the other side, we also can retrieve other kinds of biomolecules from microfossils, such as proteins, lipids, and sugars. And we know that actually they are better preserved over time than DNA, and can give us a clue into the, for example, metabolic pathways and how those have changed in certain organisms in response to environmental change. And then final advantage that I would like to mention here, and probably it's not the only one that microfossils have, is that they can provide genome diversity. What does that mean? It means that for some taxa, it is possible to retrieve microfossils that belong to different stages of their life cycle. In case of, for example, tree, we can get leaves or roots or bark, and that represents the vegetative phase. And then we can also get pollen and seeds, which represent the reproductive phase. And then by extracting the DNA from these different life stages, we can actually more comprehensively understand the response of a certain population of a certain taxa to environmental change. Okay, so these are all the advantages. Now, in terms of the feasibility, so the idea of microfossil DNA is not a new one by any account. So the original study, one of the oldest ones that at least I'm aware of, dates back to 1996. and it was done by Suyama and co-authors, and they retrieved a short chloroplast DNA sequence from a conifer pollen dated to 150,000 years ago. And so they were also the first ones to say, to propose that by analyzing fossil pollen DNA, we can identify plant species in the paleo records. And then there was a bit of a gap in the fossil pollen DNA research till about mid-2000s, when there was this paper from Parducci. And this was the first study that went beyond just taxonomic identification idea and actually demonstrated that it is possible to track population dynamics using fossil pollen DNA. And they did this for Scots pine, Pinus sylvestris. They took pollen grains from a single site in lake settings that were 110,000 years ago. They successfully extracted DNA, and they showed that there was a population continuity at that site through 10,000 years. But despite all of these mentioned advantages and demonstrating feasibility, there has only been about 10 or less microfossil DNA studies to date, even though the idea, as I mentioned, it spans 30 years. In contrast, as I mentioned before, there's well over 100 sedimentary DNA studies. So then there's a question we have to ask, and that is why so few studies have used microfossil DNA. I would argue that the biggest challenge is methodological in nature. So there are significant technical challenges in working with microfossil DNA. And to illustrate that, I'll show you what a standard eDNA sequencing workflow looks like. So we start from a sediment material, such as a sediment core. We then subsample, and once we have our sediment subsamples, we move directly to DNA extraction. After DNA extraction, we then prepare the DNA for sequencing, and this can be done in different ways. Here at GeoGenetics, we regularly do genomic library construction. And then after that, we move to bioinformatics workflow that includes quality control, mapping to do taxonomic assignments, and a DNA authentication to confirm that it's ancient in origin based on damage patterns. But if we want to do a microfossil DNA sequencing workflow, we cannot go directly from sediments to DNA extraction. We have additional steps, and they're not trivial. So we first have to isolate the targeted microfossils from the sediments, and before that, we have to identify them, we have to find them. And then once we get a clean fraction of our targeted microfossils, we have to sort them, meaning that we need to move them into individual reaction chambers where we're going to extract the DNA. Otherwise, we wouldn't get a single microfossil DNA, but multiple. And then after that, we need to lyse each of these separate microfossils. And for the lysis, we need to basically break open microfossils and get the DNA accessible. And then there's finally an additional challenge of actually extracting DNA because we are dealing with a considerably smaller amounts than we would when you start with a chunk of sediment. But there is a methodological proof of concept, and it comes from Bennett and Parducci from 2006. So what they did, and I'll just explain briefly, so they would basically prepare manually micropipettes by extending regular glass pipettes over flame. And then they would take those micropipette kind of tips, and then they would go into a sediment sample and extract individual pollen grains, one by one, all manually. Then they would place them in a small tubes, such as PCR tube, and then they would have to go under the microscope and find again that pollen grain there, and then aim at it with that micropipette again to break it open. As you can imagine, for each of those pollen grains, that's a lot of work. And also, they worked with quite large pollen grains, close to 100 micrometers. But there's many pollen types and other types of microfossils that are considerably smaller. So for those, it virtually would be impossible to try to open them and break them with a micropipette. And then after all this laborious work, they also found that the success rate was quite low. So they worked with 1,000, 1,500 grains. And out of all of those they processed, only 1.5% yielded DNA. So as you can imagine, if a microfossil DNA should be uptaken as a method, we have to find a way to actually screen grains and process grains faster or in a more automated way. So several years ago, when we started this line of research, we decided that to do that, we will try single-cell sequencing technologies. So back then, those were emerging technologies. Today, they're much better established. There are many different single-cell sequencing technologies out there. I don't have time to go over even most of them, but I will just note that they all share the same main principle, and that is that they are aiming to be able to retrieve and process genomic material from individual cells at scale, meaning hundreds to hundreds of thousands of cells in parallel. And so they do that, they achieve that in different ways. So, for example, there are microwells out there where they're just big enough to put one cell in there to process it. Another approach is microfluidics, where very small nanoliter volumes of liquid are manipulated to encapsulate cells and sort them out. And then also an interesting approach is in situ, where the cells are not moved in a reaction chamber, but the cells themselves are used as the reaction chamber in which you bring in the reagents and you process DNA in there inside. So when we were starting this line of research, we were actually quite optimistic. We thought that given all this wealth of technologies, we'll certainly find some that will work well on microfossil, maybe even too many to choose from. But we were very wrong because we overlooked that microfossils have some characteristics that make them quite different from cells. First of all, the single-cell sequencing technologies are developed with cell culture samples in mind, meaning that the sample is uniform. All cells are of the same type, the same size, same shape. But if we want to work with microfossils, even a certain type, such as pollen, in a sediment sample, we get many different shapes and sizes, as you can see here. And then also, cells tend to be smooth. They also tend to be light, so they don't sediment fast. And the opposite is true for the microfossils. Because they have mineral shells, they sediment faster. And also they have all sorts of patterns and even appendices on the surface that makes them easy to clump together and so difficult to separate. And also because of these hard shells, those shells are difficult to actually break open. While with the cell membranes, you can actually easily dissolve them through enzymatic activity or some weak detergent. And a nice example of a technology that we were very interested in adopting, but it was just not compatible with microfossil characteristics, is droplet microfluidics. And this is an approach where single cells are encapsulated in aqueous droplets. And you can see them here. And so it's one cell per one droplet. And so in this process, we are creating the droplets and they are immersed into a carrier oil that makes them stable. And it's a beautiful process. It's actually very easy to scale up to produce thousands or even tens of thousands of droplets with single cells inside within minutes. So it's very high throughput. And we have tried this on pollen and it worked really well. So we used the kind of chip you can see here in right, where we have two tubes going in. One tube is bringing the sample, the other tube is bringing the carrier oil. And so then the droplets are being created within the chip. And then there's an outlet that then we can collect the droplets at the end of it. So that's all great. But then the next step after that is lysis. And for droplets, they are too sensitive to be able to handle a strong acids or strong, any kind of mechanical force. So they can only stand enzymes and detergents. but those actually do nothing to most of the microfossil shells. So the lysing is actually not possible to do in droplets. So over the years, we have tried many different systems. And I don't have time to actually show you all, but I will note that I'm very happy to say that recently we have managed to establish a prototype of the first high throughput workflow for ancient pollen DNA analysis here at Geogenetics at Copenhagen. And so the central feature of this workflow is a semi-automated microscopy-based single cell sorter instrument that is modular, meaning that you can add multiple modules onto it. The two modules that we are using, the first one is microcapillary module that you can see on the right, and it consists of a syringe with a microcapillary, and the microcapillaries come in different sizes, ranging from 20 micrometer to 160, so you can cover many of the microfossils of interest in that range. And you can see a rim of that microfossil in the right bottom corner, where it's basically going over a pollen grain, and then we can suck up the pollen grain using a small volume, which means that we will not catch the other unwanted particles around it. And then we can move it to the reaction chamber. Following that, of course, is the lysis, which has been one of the major issues for the workflow. But there's another module on this instrument that uses UV laser. It's a weak UV laser, but it is strong enough to break open cells, and we have tested it on pollen. And as many of you know, it's really hard shell, and it works also well. And you can see that in the top left corner. So, in short, we are still in the early stages of microfossil DNA workflow development. There's many steps we need to perfect, but we are very excited. We feel that this instrument has a huge potential. And even though our focus at the moment is on pollen, we will also eventually apply it to other types of microfossils because it should in principle work. Okay, and I will just note in the end that this research is being funded by the new Center of Excellence for Ancient Environmental Genomics. And it has been basically established to the funds from the Danish National Research Foundation. As part of this center, we work on many different method developments, not just the wet lab, but also dry lab. And we are also applying ancient environmental DNA methods to answer very different questions, from the resilience of wild ecosystems to evolutionary histories of humans and pathogens, all the way to trying to develop more resilient crops to the upcoming climate change. We are very interested in collaborating with scientists across different disciplines. So if any of this research seems like a fun and interesting research to be part of, please do reach out. And I would like to leave you with the following thought, and that is that there is an immense value of using different paleoproxies in our studies. And ancient DNA is a valuable addition. But to move things forward, it's always worth looking at other sources of information about the past. So in case of ancient DNA, we have well-established macrofossil methodology and ancient environmental DNA. But I hope that I have convinced you that microfossil DNA represents also an excellent opportunity to expand our paleo toolkit. Okay, I'll stop here. Thank you for your attention. And just wanted to, of course, acknowledge the fantastic researchers that I had the opportunity to work with over the years.
All right. Thank you very much. That was super cool. So everybody, just so you're aware, the chat is now open. If you have any questions for Anna, please feel free to type those into the chat. Make sure that you tell us where you're from and make sure you share it with everyone, and we can get those questions answered for you. But I have a question. So how robust is the DNA? How easy is it to break down? Or does it kind of just like to stay there?
Yeah, no, it's not very robust. Because as soon as an organism dies, already within the organism, the nucleases, which are normally separated, and they are the ones that are starting to break down DNA, they are in a living cell separated from the DNA that we want to preserve. But as soon as the organism dies, those nucleases start to degrade DNA. So that's already a process that starts. And then when the DNA comes into the environment, there's also a lot of nucleases out there produced by viruses and bacteria, and they continue that degradation process. And then you also have water and sunlight. They're also, speeding up, catalyzing that process. So really, so far, we have limited evidence, but the evidence we had suggests that it is key that DNA attaches to something that protects it. So either mineral particles or humic acids or something like that. If it's just kind of free in the environment, it will not survive for a long time.
Cool. All right. Looks like we have our first question. This is Amelia from Utrecht. Are there particular sequences that are more stable? Does the basis arise because of that? Is there actualistic taphonomic studies to check if we selectively losing some sequences?
Yes, we don't have enough information to be sure. So this is ongoing work, but there is some evidence to suggest that certain sort of nucleotide combinations are more resilient than others. So, but that would be on a really small scale, because of course we don't get really long parts of the genome, except for some of the repetitive ones that would be particularly sensitive or particularly strong. So I don't think that on the scale of the whole genome that we would be very much skewed. But as I say, it's a work in progress.
Right, the next question is from Ross. If anyone can identify a microfossil, what are the advantages of being able to characterize its DNA?
Right, fair enough. Well, I'll take pollen as an example. So we can identify, when we see a pollen, what genus it is in Europe, for example, very easily. More difficult in tropics where diversity is higher. We can rarely look at a pollen grain and say it is this specific plant species. But when we want to start looking at specific population dynamics, we want to know whether we're dealing with Betula nana or Betula pendula, right? There's differences there. And we cannot, it's very difficult to do that morphologically. So this is where extracting DNA and confirming whether it's one species or the other is really helpful. But then also what we're interested in is not just knowing what species are there, but also what was, how did they react to changes in the past through their genetic variation? What were the adaptations? What was the population size? How does that change? And this is very difficult to get from microfossils alone. Pollen abundance can approximate the amount of maybe that taxon landscape, but there's also been some work showing that that's not always the case. So yeah, and especially to, for example, look at selection. So changes in the specific genetic variants over time in response to certain changes in the environment, that is virtually impossible to do just through the morphology.
All right. Our next question is from another Anna in the UK. Will the optimization of successfully retrieving microfossils for eDNA analysis lead to the improvement of reference databases by incorporating less studied or previously unknown species? Will this in turn facilitate the ability to accurately identify and classify species directly from sediment samples?
Yes, that is one of our hopes. It is not going to be an easy task because the amount of DNA we have in a microfossil is very low. So the chances that, so we have been talking about single genomes, but they would not be genomes that have very high coverage. We would have many missing parts of it getting from a single microfossil. So identifying species that are unknown to science at the moment, that went extinct and we don't know about them today, it will be quite challenging to do, but not impossible. And definitely we can, for example, use macrofossils, so the larger fossils, to do something like that. And it's actually some work is being done already now on some extinct megafauna, right? But yeah, I wouldn't exclude the possibility, but it will be a challenge.
That would be very exciting.
It would be super exciting if we were able to do that, though.
Amelia, again, how often do you get mysterious sequences that you cannot match confidently with anything known? And what do you do with them?
Okay. Yeah, that happens. And you know, the more diverse ecosystem is from which you are retrieving DNA, the more likely you're going to have that. Because, of course, how well we can match the sequences depends in large part of how good our genome reference databases are. And they are not excellent at the moment. They are improving every day because there's more and more reference genomes that are being created and added to these databases. But yeah, because of that, we can have reads that don't match to anything. We can have reads that match to one taxa when they possibly belong to another taxa that we don't have their genomes in the reference database. So yeah, these are all challenges that we are dealing with on the bioinformatics side. I wouldn't be able to tell you, give you a number of how many times or what is the percentage of time that happens. But yeah, it definitely happens.
All right, the next question is from Stephen. Sediments contained in paleo-beaver pond environments often undergo wet drying cycles and experience redox conditions. Would this technique be suitable and produce reliable data to be used for the reconstruction of Holocene terrestrial forest composition that was deposited over the paleo-beaver pond depositional environment?
Good question. So these are layers that were continuously going from wet to dry cycle, if I understood correctly. So this is not ideal, because what you would like is the perfect case scenario is where there's a deposition of DNA in certain conditions, and then those conditions remain the same throughout until you actually collect that sample and start processing it. Having drying and wet cycle can cause potentially some leaching of DNA through the layers, so you might be losing the robustness in chronology. And you are also exposing the DNA to these changing conditions where it can start degrading because it's getting dry and wet and dry and wet. So it is not ideal, but not impossible. But from the question of whether you could then use the DNA to understand the paleoclimatic or paleoecological dynamics, that you can certainly do. If you can get viable DNA from those layers and you can confirm that they really belong to that time period, then yeah, that's possible to do.
All right, well, I think maybe we'll start wrapping things up now. If anybody else has any further questions, I'm sure they could send you an email and ask you those, or maybe some potential collaboration out there.
Yes, fantastic.
And,
yes.
Just thank you again for an awesome presentation. Make sure that everybody checks out our website so you can see what next month's seminar is going to be about. It's going to be at the regular time, 4 p.m. UK time, where we will learn about taphonomically active zones and skeletal preservation. So that should be also very interesting. So, thank you again, Anna.
Thank you, Simon.